So let's get started. It is my greatest pleasure to introduce Dr. Gopal Sapkota. And Gopal is originally from Nepal, but obtained a master's degree in biochemistry from the University of Bath in UK. And then he received his PhD from the University of Dundee in 2003, and then joined John Massague's lab at Memorial Sloan Kettering Cancer Center in New York to undertake postdoctoral research to study the TGF-beta pathway regulation, for which he received the prestigious Damon Runyon Cancer Research Fellowship. And Gopal returned to Dundee in November 2008 to start his own group at the MRC PPU, and in 2014 became a tenured program leader. And today he's gonna introduce an amazing induced proximity driven phosphatase dephosphorylation. And thank you for accepting our invitation. And now the floor is yours, Gopal.
Okay, thank you very much, Hozung, for the invitation and the committee. And let me just, okay. So it's a great pleasure to be presenting this, sharing our work with you. And today I think I'm going to talk about targeted dephosphorylation of phosphoproteins through induced proximity. So protein phosphorylation is fundamental in every cellular process, and the process itself is reversible. So protein kinases, there are roughly about 500 of those that mediate phosphorylation of proteins, and protein phosphatases dephosphorylate the proteins. The process itself is tightly controlled, and the disruption of the phosphocontrol is a hallmark of many diseases, including cancer and neurodegeneration. And the importance of this family is highlighted by the fact that there are now over 80 kinase inhibitors that are in clinical use, and many more that are in advanced clinical trials. The problem is the reverse, which is the phosphatases have been very challenging to target for inhibition or activation. And that's primarily because there are limited number of phosphatases, 200 compared to 500 plus kinases. That means they are more promiscuous. And by far, PP1 and PP2 family of phosphatases that are holoenzyme complexes, meaning there are multiple proteins that make up the active components. They are known to dephosphorylate over 90% of all phosphoserine and threonine residues. And the substrate specificity on them comes from these so-called regulatory subunits. But there are some apophosphatases, such as PPM1H, that are thought to be more selective. In clinic, there are virtually no activators or inhibitors available at the moment. And another challenging issue with phosphatases is many of the substrates remain poorly established. So the rationale for substrate level phosphocontrol, when we first started working on this, is as follows. So you can have any protein that is phosphorylated on multiple residues by different kinases. In this case, let's assume kinase X phosphorylates one side and kinase Y another one. You can imagine different phosphorylation sites could actually have distinct biological outcomes. And if you want to inhibit kinase X, the problem is many kinases have multiple substrates. So you could have many other phosphorylation events that are completely blocked if you block this kinase. Similarly, if you block kinase Y, you might have many different other substrates that are also inhibited. And if you want to inhibit the phosphorylation of both, then you'd have to inhibit both kinases, and that adds to the list of all these off-target effects. So if you were able to selectively recruit a phosphatase that dephosphorylates either this or both of them, then you bypass all of the effects of kinase inhibition. So achieving a substrate level phosphocontrol by redirecting phosphatases would be much more desirable than inhibiting kinases upstream. So how do we go about doing that? I think we can take a lot of examples of targeted degradation that's been established, and it's kicked off. So we need a bifunctional element which has a phosphatase or a phosphatase binder that recruits the phosphatase of interest, and then the protein of interest that element that binds to the protein of interest and induces proximity between the phosphatase and the protein of interest. And the hope is if there's a ternary complex between these, that some of the phosphates will be removed by the phosphatase and you result in targeted dephosphorylation, and hopefully that will lead to changes in downstream signaling. So we've taken several approaches. The first one I'm going to talk to you about is mainly done by past students, Luke Simpson and Luke Fulzer. And we need this approach in affinity-directed phosphatase system, or the add phosphatase system. What it is it has a phosphatase or phosphatase binder element linked to a protein of interest binder, and it's a polypeptide that we decided to use on the phosphatase binding element the catalytic subunits of PP1 A holoenzyme complex, and also had a negative control with the phosphatase dead mutant. We also included PP2A and the dead mutant, and this is because these are known to be the most promiscuous phosphatases that are there. So for the protein of interest binder, we actually utilized a antigen-stabilized anti-GFP nanobody that was developed by the Sepco lab, where there are 6 mutations in this nanobody that make it unstable until and unless you have the GFP moiety in cells. So the useful approach basically of this nanobody is when you have a GFP knock-in into your target protein, then that can stabilize only the binding pool of the phosphatase that is bound to anti-GFP, but it does not allow for the phosphatase overexpression that might actually have detrimental effects. So using this system, we basically package the phosphatase, PP1 or PP2CA, with the anti-GFP 6M nanobody, package them in retroviruses, and for protein of interest, we knocked in a FAM83D, which we work on in the lab. So what is FAM83D? FAM83D is a scaffold protein that delivers CK1 alpha in the mitotic spindle. So what you can see here is in FAM83D GFP knock-in cells, it localizes to the mitotic spindle of STLC, basically induced mitotic arrest cells, where you see radial alignment of the chromatids, and this is the spindle assembly where FAM83D localizes to. It recruits CK1 alpha to the mitotic spindle, as you can see with overlapping staining here. And in FAM83D knockout cells, of course, you don't have FAM83D, but CK1 alpha does not localize to the mitotic spindle either. In the wild type cells, you don't see the GFP signal, but you can see that CK1 alpha is localized to the mitotic spindle. And one other thing happens in mitosis. So FAM83D is heavily hyperphosphorylated in mitosis, and it interacts with CK1 alpha only during mitosis. So you can see that the co-localization of FAM83D and CK1 alpha in mitosis starts at prometaphase, and by telophase and cytokinesis, you start seeing degradation of FAM83D. So we know that FAM83D is phosphorylated in mitosis, but we do not know what the role of mitotic FAM83D phosphorylation is. So we decided to use the add phosphatase system to deliver either PP1 or PP2CA to GFP knocked in FAM83D in cells. And we used wild type cells as negative controls. So what you see from here is in asynchronous or mitotic cells, in the wild type background, you see a heavy phosphorylation of FAM83D in mitosis. Taking FAM83, so taking anti-GFP PP1 or anti-GFP PP2A in the wild type background that do not have GFP FAM83D, they do absolutely nothing to the phosphorylation, mitotic phosphorylation of FAM83D. But now, if you look at GFP FAM83D knock-in cells, in control cells, you see that mitotic band shift that's caused by phosphorylation. But now both PP1 and PP2A almost completely dephosphorylate this mitotic band shift in mitosis. So we basically can co-localize all of these constructs to the mitotic spindle with FAM83D in those cells, suggesting that the binding of GFP to the anti-GFP is taking all of the phosphatases or the mutants into the mitotic spindle. So the critical experiment we did is this one here. We basically pulled down flag using anti-flag antibodies, anything that was bound to these phosphatases. And what you can see is in the control nanobody alone, you can see mitotic FAM83D that is phosphorylated, that is pulled down. But with the wild type PP1 or PP2A, you see completely dephosphorylated FAM83D coming down, whereas the same FAM83D pool that is pulled down by the phosphatase dead mutants is still phosphorylated to the same level as the control nanobody, suggesting that this dephosphorylation that we see is definitely down to the catalytic activity of the phosphatases that we deliver to the mitotic spindle. So I told you that the phosphorylated FAM83D is degraded upon mitotic exit, and it turns out that the targeted dephosphorylation of FAM83D in mitotic spindle actually stabilizes the FAM83D that is dephosphorylated. So you no longer, the rate of degradation of FAM83D in mitosis or upon mitotic exit is severely delayed by dephosphorylation, suggesting that the phosphorylation of FAM83D in mitosis is a signal for its degradation. We also did a unbiased phosphoproteomic analysis using either the anti-GFP PP2A wild type or the catalytically dead mutant. And what you see is FAM83D is one of the top sites that's dephosphorylated, and there are only very few sites that are dephosphorylated. And among these, most of these are actually spindle localized proteins in mitosis. And there were very few proteins that actually changed abundance when we did the total proteomic analysis. So the add phosphatase system we also tested other substrate, including tau. And tau is basically a microtubule binding protein, and it's hyperphosphorylated in Alzheimer's disease. And the hyperphosphorylation is thought to lead to its dissociation from microtubules. And this basically allows for aggregation of tau into neurofibrillary tangles, basically. So again, we used a very similar approach and asked, could we target dephosphorylation of phospho-tau using the same add phosphatase system? And could that affect microtubule binding and prevent tau aggregation? So the approach is very similar. So we packaged this time PP2CA with the anti-GFP nanobody. we knocked in SK-NMC cells on tau with a GFP tag, and then basically transduced the retroviruses encoding PP2CA anti-GFP6M add phosphatase to see what it did. And what you see from this figure is, if you just focus on the middle two lanes, is when you have anti-GFP wild type or catalytically active phosphatase, you see complete dephosphorylation of most of the sites, phosphosites that we tested. With the phosphatase dead, you don't see. With all the other controls, just the phosphatase, just the anti-GFP, or no treatment whatsoever, you see the same levels of phosphorylation. Next, we also did immunoprecipitation of the phosphatase or the catalytically dead phosphatase, and then looked at what happens to the bound tau phosphorylation. What you also see is the phosphatase dead mutant basically does not affect the phosphorylation of tau on all these sites, whereas everything that is bound to the wild type phosphatase is completely dephosphorylated. Again, suggesting this is really a targeted dephosphorylation of tau. So we do see a robust targeted dephosphorylation of endogenous tau GFP by PP2A. But what is the extent of dephosphorylation? So what we did is we used these two again, immunoprecipitation of flag to immunoprecipitate bound tau that is bound to wild type or the dead phosphatase, and then did a global proteomic and phosphoproteomic analysis. And what you see is there are many tau phosphosites that are actually dephosphorylated. And in all, we see 14 tau phosphosites that are targeted for dephosphorylation. So the question was, does the targeted dephosphorylation of phospho-tau affect microtubule binding? And we did a very simple microtubule binding assay with immunofluorescence done by Tyrell Cartwright in the lab. And what we see is in the cells that express the anti-GFP6MPP2CA, you see occupancy of GFP tau on microtubules that overlaps with the tubulin staining, but in the phosphatase dead mutants, that was severely reduced. And this is the quantification of the data, suggesting that targeted dephosphorylation of tau enhances the microtubule binding of endogenous tau GFP in these cells. So the summary of the add phosphatase system is basically we've shown a proof of concept for efficient and selective targeted dephosphorylation of FAM83D and tau that is achieved by either PP1 or PP2A. So targeted dephosphorylation, I showed, impacts the function of the proteins. And using the same system, we basically use this for targeted dephosphorylation of a whole range of different substrates by the add phosphatase system. And we've also identified new phosphatases that can be redirected to dephosphorylate different substrates. But the key limitation with the add phosphatase system, you might have already noticed, is when you introduce GFP, anti-GFP tagged phosphatase in cells to the GFP tag target protein, the binding is constitutive. So if the binding affects the function of the protein that is independent of the targeted dephosphorylation, there's no actual way of separating this from the phosphatase here. So we basically asked, could we develop a small bivalent molecule that can do pretty much the same thing, but then avoid the constitutive nature of binding so that we can actually look at the system in more depth. So this is where there have been several other proximity-inducing molecules for different tag systems, including halo-based system. but that Craig Cruz's lab actually published, and we also developed. But in our hands, that system has not worked very well for the targets and the phosphatases that we have actually used. So we decided to go for another tag system that did not use a covalent binder. And for this, we conceptualized that we would use a D-tag system on a target protein of interest and a bromotag system for a phosphatase of interest. And these you could knock in with CRISPR. And then we developed a bromotag D-tag proximity inducing chimera, again, harnessing the very well-established molecules from Nathaniel Gray and James Bradner's lab, using the D-tag ligand here, and Alessio Ciulli's lab in Dundee using the bromotag ligand that has been established and used in PROTACs. So we made this molecule, and if the molecule actually works, then what we would expect is a proximity induction between the D-tag and the bromotag with the POI and the phosphatase. And if you get a ternary complex forming, then you'd expect the phosphorylation to disappear on the target protein, and hopefully that leads to consequences downstream. So that was the idea. So we decided to use two different target proteins, initially TFEB and SMAD3. And then for phosphatases, we decided to explore a few of the phosphatases that had already worked for us in the AdPhosphatase system. So this work was mainly led by PhD student Abby Brewer and postdoc Feng Zhao. So initially, TFEB is a transcription factor that basically, in response to starvation, exercise, or stress, that either activate AMP kinase or inhibit mTOR, essentially lead to the dephosphorylation of TFEB that then translocates into the nucleus. And in the nucleus, it basically transcribes a lot of genes that regulate autophagy and lysosomal biogenesis. So you can see that under normal conditions, TFEB is heavily phosphorylated. And this can be visualized in a simple Western blot. Basically, you can see that this band migrates slightly higher compared to the lysates that are treated with lambda phosphatase that migrates much further down. And you can see in DMSO-treated cells, you get this higher molecular weight band, whereas if you activate AMP kinase by MK8722, that leads to inhibition of mTOR, you see a downregulation of this, so sort of the mobility shift downwards of this band, suggesting TFEB is dephosphorylated and it translocates to the nucleus. Similarly, if you inhibit mTOR by taurine-1, you see a downward shift. So we use this shift as a basically decoy or as a reporter for levels of phosphorylated TFEB in cells. And so we used a D-tag TFEB with the bromotag PP2CA. And then the idea is if you add the BDPIC bivalent molecule, then you'd actually induce proximity and then see what happens to TFEB. So the first experiment we did was with the wild-type phosphatase that was bromotagged and a phosphatase dead mutant of PP2CA with the TFEB that was D-tagged. What you can see immediately is 100 nanomolar TFEB in 24 hours. You see a robust downward mobility shift of TFEB. This does not happen with the phosphatase dead mutant. So we took the lowest concentration, 100 nanomolar, and then did a time course. And here we also included no phosphatase control, just the bromotag. Again, there's no dephosphorylation or mobility shift of TFEB. With the wild-type phosphatase, you can see a robust downward mobility shift within 2 hours. With the phosphatase dead again, there's absolutely no change. So we actually took this a bit further and went to minutes. And what you can see is within 5 minutes, 100 nanomolar BDPIC induces a robust dephosphorylation of TFEB. So it's a really fast-acting molecule that induces proximity basically between these two and then causes dephosphorylation of TFEB. So it is completely reversible. So we did a washout style assay where we induce the dephosphorylation for 1 hour and then wash off the molecule and basically look at what happens to the phosphorylation. Within 1 hour, the phosphorylation of TFEB completely comes back. We switch the tags. So we put bromo tag on TFEB and D tag on PP2CA, but the outcome is exactly the same. So BDPIC still induces dephosphorylation of bromo tag TFEB this time, and the phosphatase dead or the D tag alone do not do it. So we compared the natural sort of ligands that induce dephosphorylation compared to BDPIC. And what you see is BDPIC not only matches the natural dephosphorylation that you see, but you also see additional mobility shift downwards, suggesting that there are multiple other phosphosites in TFEB that have not been actually characterized yet. So we also did a simple pull-down assay where for one of the tags, bromo tags, we had a HA tag. So we pulled down the bromo tag in the absence or presence of BDPIC to look for TFEB. And what you see is only in the presence of BDPIC do you see interaction between the bromo tag phosphatases and the D tag TFEB, suggesting that it is actually forming a ternary complex. And what's also really promising is everything that is bound to the phosphatase is dephosphorylated TFEB as well. So we also did competition assays using the warheads that were used for bromo tag and D tag, using the negative protags, D tag V1 negative and cis AGB1. So these are the inactive protags for D tag and the bromo tags. And when we use that, you can see that the BDPIC-induced dephosphorylation is completely rescued in the presence of D tag V1 or cis tag AGB1, suggesting that it was really the bringing together of these two components that was causing the targeted dephosphorylation in the first place. And I mentioned that, as you can see from this figure, so when you add taurine to cells, that causes dephosphorylation of TFEB, and with that, it causes nuclear translocation of TFEB in all conditions. But when we use BDPIC, it only causes nuclear translocation of TFEB in the presence of wild-type phosphatase, but not in the presence of just the bromo tag or the phosphatase dead mutant, suggesting that this targeted dephosphorylation by BDPIC was sufficient to cause nuclear translocation of TFEB. And in the nucleus, we also looked at transcription of TFEB target genes upon targeted dephosphorylation of TFEB. And what you can see is you do see a moderate increase in the transcription of target genes upon BDPIC treatment, only in the presence of wild-type phosphatase, but not the phosphatase dead mutant or the bromo tag alone, suggesting that the nuclear translocated TFEB upon BDPIC treatment is transcriptionally active. And we could do all of this when we actually knocked in the same cells that had D tag TFEB knock-in. We were able to introduce a bromo tag on one of the alleles of endogenous PP2CA. And you can see that BDPIC induces this targeted dephosphorylation as well as nuclear translocation of TFEB at the endogenous level. So in this case, I think you could really redirect endogenous PP2CA to dephosphorylate TFEB using BDPIC induced proximity. We also took these cells and actually did a unbiased total and phosphoproteomic analysis. This was done by Gazanan. And what we saw is two phosphosites on TFEB that were downregulated in the wild type, so in basically BDPIC treated cells. And these are shown here. And of course, with the unbiased endogenous phosphoproteomic, it's the sensitivity that determines what sites you identify. And then another encouraging thing was there was no global dephosphorylation or heavy phosphorylation caused by the molecule itself. So we also tested targeted dephosphorylation of SMAD3, which is a completely different transcription factor that behaves in a completely opposite way. In this case, so it responds to TGF-beta ligands by being phosphorylated by type 1 receptors, TGF-beta receptors, and it's the phosphorylation that causes nuclear translocation and transcriptional activation of SMAD3. And in the nucleus, it can transcribe genes, many hundreds of genes, including PI1 and SMAD7. And one of the functional readouts of this is TGF-beta causes epithelial to mesenchymal transition, and that can be measured as well. So what we can see is when you add TGF-beta to cells, within 1 hour, you see a robust phosphorylation of SMAD3 at this tail phosphosites, and it naturally gets dephosphorylated after a certain time. So in this case, we also looked at the natural rate of dephosphorylation by a washout style assay. So we basically induced the phosphorylation, optimal phosphorylation for 1 hour. And then we washed out TGF-beta from the media and then followed over time the natural rate of dephosphorylation. We did this because if you want a targeted dephosphorylation, then any targeted dephosphorylation has to really exceed the natural rate of dephosphorylation. So for this one, we basically picked a 2-hour time point where we roughly see 50 to 60% of the natural sort of phosphorylation, natural rate of dephosphorylation compared to the controls. So in this case, we chose a range of phosphatases, 4 different actually, ppm1A-H, pp2CA, and pp1CA, and did a washout style assay. So time 0 is optimal phosphorylation. 2 hours after washout is the natural rate of dephosphorylation. So any dephosphorylation we see after addition of BDPIC would have to exceed this particular rate of phosphorylation. And here you can already see that ppm1A causes slightly more dephosphorylation, ppm1H causes even more. Whereas pp2A and pp1CA did not really cause dephosphorylation to a great extent in terms of SMAD3 tail phosphorylation site. So we took ppm1H forward because we saw the most dephosphorylation with ppm1H, and we were able to demonstrate that the targeted dephosphorylation of SMAD3 by ppm1H was activity dependent because the catalytically dead mutant of ppm1H did not cause additional dephosphorylation. So we knocked in DTAG SMAD3 with bromoTAG ppm1H in cells, and what we were able to show is you can cause dephosphorylation by BDPIC, and this dephosphorylation can be completely rescued by cis-AGB1, which again has the negative protag for bromoTAG that competes with the bromoTAG binding. And we can rescue the same thing with DTAG V1 negative, which again competes with the DTAG binding. So this really is targeted dephosphorylation of SMAD3 caused by recruitment of bromotag ppm1H. We were able to demonstrate that in pull downs, BDPIC induces an interaction between SMAD3 and ppm1H, and that interaction can be competed out again with cis-AGB1, suggesting that the binding is really inducing proximity to the two components here. So then we basically did some functional analysis at the endogenous level, where A509 cells that were knocked in with bromotag on ppm1H with CRISPR and DTAGs on SMAD3. And in these cells, you can very clearly see that the natural rate of dephosphorylation here seen is basically more than the levels of phosphorylation of SMAD3 that is caused by BDPIC. And under these conditions, you can also see one of the downstream targets of TGF-beta, Pi1, is reduced in these cells. We also looked at transcription of Pi1 and SMAD7. And what you can see is compared to controls in TGF-beta treated cells, the levels of the transcripts are much lower, significantly lower in those cells that are treated with BDPIC. So we also actually, I think going back to phosphorylated tau again, we also tested the targeted dephosphorylation of tau by BDPIC using the same system of tagging bromotag on one end and DTAG on the other. And this was done by Gazanan. And what you see here is in untreated cells, you do not see any dephosphorylation. In cells that are treated with different concentrations of BDPIC, in the wild type DTAG PP2CA expressing cells, you see a robust dephosphorylation of multiple sites on tau, whereas in the phosphatase dead expressing cells, you do not see any added dephosphorylation in response to BDPIC. We can do this at the endogenous level where tau is tagged with bromotag with CRISPR, and one of the alleles of PP2CA is tagged with DTAG. And what you see is the phosphorylation or targeted dephosphorylation of tau on this serine 396 is dose dependent on BDPIC and also time dependent, because you can see within 2 hours, you see a robust dephosphorylation of tau on phosphoserine 396. So we're actually doing unbiased phosphoproteomic analysis on these, and we should be able to identify multiple phosphorylated sites on tau that are targeted for dephosphorylation. So, and we still are planning some functional consequences of targeted dephosphorylation of tau using the BDPIC model in these. So in summary, I think I've been able to demonstrate that we can efficiently and selectively target dephosphorylation of TFEB and tau by PP2CA and SMAD3 by ppm1H using the BDPIC molecule. And the targeted dephosphorylation impacts protein function. And of course, I think we are actually moving forward and trying to develop phosphates that do not rely on these tags anymore, but basically trying to recruit, develop ligands that recruit and not inhibit phosphatases that could be utilized to dephosphorylate endogenous targets as well. And this is an ongoing collaboration with Alessio Ciulli in Dundee, Adrian Soren in Dundee, and then Amgen as well. But the key bottleneck still is there are no good phosphatase ligands. There have been 1 or 2 reports of endogenous ligands for phosphatases, but we're still yet to actually confirm that they are fit for purpose in our systems. So the other, I think, advantage that I just would like to actually expand on is the BDPIC that we have developed. I mean, does not have to be limited to phosphatases. You can basically imagine any enzyme of interest that can be knocked in with a bromo tag, any protein of interest that can be knocked in with a D tag, and within 5 minutes at 100 nanomolar, you can actually induce proximity and then start testing the functional consequences of targeted post-translational modification of the proteins of interest, or even generally what happens when you recruit two different proteins together inside cells. So this would basically allow us to test really new drug modalities. Okay. With that, I'd like to acknowledge my lab. I think I did acknowledge during the talk most of the people that were involved in specific aspects of those. For chemical synthesis, I'd like to acknowledge Natalia Safiro, who's retired now. We have a lot of support in Dundee that provide us with a lot of technical help. And I'd like to acknowledge collaborators, Keith Sakamoto from Novo Nordisk, who was involved with the TFAB project, Dario and Rotimi for providing us with the PPM1H knock-in cells, the mass spec service within the MRC PPU. FOSTA collaborations, I mentioned Alessia and Adrian, and then Callum and then Ria are driving those projects. And I guess I'd like to acknowledge a lot of the funding that we receive from different sources. And special mention to Bainam and Nathaniel, who were former Dana-Farber colleagues that basically have been very generous with all the reagents and advice over the years. And